HIV Life Cycle by Erica Howard. Step 1. Binding and fusion. HIV begins its life cycle when it binds to a CD4 receptor and one of two co-receptors on the surface of a CD4 plus T cell, as shown here. The virus then fuses with the host cell. After fusion, the virus releases RNA, its genetic material, into the host cell, as shown there. Step 2. Reverse transcription. An HIV enzyme called reverse transcriptase converts the single-stranded HIV RNA to a double-stranded HIV DNA, as shown there. Step 3. Integration. The newly formed HIV DNA enters the host's nucleus. This is where an enzyme called integrase hides the HIV DNA within the host cell's own DNA. This integrated HIV DNA is called provirus. The provirus may remain inactive for several years, producing few or no copies of HIV. Step 4. Transcription. When the host cell receives a signal to become active, the provirus uses a host enzyme called RNA polymerase to create copies of the HIV genetic material as well as shorter strands of RNA called mRNA. The mRNA is used as a blueprint to make a chain of long HIV proteins, as shown there. Step 5. Assembly. An HIV enzyme called protease cuts the long chains of HIV protein into smaller individual proteins, as shown here. As the smaller HIV proteins come together with copies of HIV's RNA genetic material, a new virus particle is assembled, as shown here. Step 6. Budding. The newly assembled virus, as shown here, pushes out of the host cell's outer envelope through what is called buds, as shown here. This envelope acts as a covering and is studded with protein-sugar combinations called HIV glycoproteins. As budding occurs, the new virus steals part of the cell's outer envelope. The HIV glycoproteins are necessary for the virus to bind CD4 and co-receptors. The new copies of HIV can now move on to infect other cells. Thank you for watching.